So what was cytokinesis? It was physical division of cytoplasm, which is done at the end of cell M phase, resulting into two daughter cells at the end of M phase. We can say mitosis. First we'll talk about the animal cells. So what happens, see, suppose it's a cell, okay, this is equator or equatorial line, let's suppose it, okay, it is formed near the old metaphasic plate. Mainly in cytokinesis, it occurs by a process known as cleavage. Okay, so what happens basically, suppose this is a cell, and here it forms groove like structure. For example, wait, suppose like this, it forms a groove like structure. You can say groove like structure. We call it as cleavage furrow. Okay, and this type of this division is going on where we can call this contractile ring of microfilaments. So basically what happened here is this ring of microfilaments contractile, it will get deepened, it will get tightened, and due to this the cleavage furrow, it will get deepen, and it pulls the membrane inward around the midzone here, and thus division happens. Okay, now more thing is, see, this was a cell, wait, this was cell, okay, here it was dividing into, at this point we can say this cytoplasmic ring of microfilaments. What happened here is these microfilaments are made of actin, and furthermore this actin is associated with protein of myosin. What happened is microfilaments interact with myosin molecules of protein, which causes this ring to contract. For example, like have you seen a cylinder? You know, there is a cylinder, okay, what we can do here, we take a string, or like just take a, we can say spasm, or we can say sausage. Suppose this is a sausage, okay, we take a thread and wrap it around it, and we push the thread this side and from this side, like it divides into two, from outward to inward, outward to inward. The same happens in the animal cell, which resulting into two separate cells with different nucleus, and but different nucleus, but the same one share, or the same share of cytosol organelles and other structures. Main thing to note here, like in animal cell division, it runs from outward to inward. Now we'll talk about the plants. So in plant cells, what happens is, suppose this is a plant cell. Just for example, now here it has a cell wall. Okay, then it has plasma membrane also. The cell wall is too rigid. We cannot break it or we cannot cut it. So what happened is, the cell divides from the cytoplasm divides from cytoplasm. We can say the cytoplasm here, like it divides from inward to outward. How it is done? Basically, what happens during the telophase, the remaining of spindle microtubules reorganize into a structure which is known as phagoblast. You can say spindle microtubules organize themselves into a structure called phagoblast. Like for simple view, it is, you can say, just like this. Okay. You can say this type of structure is phagoblast. Just for explaining, okay. I'll tell you more detail in cell when we study about the cell cycle, or we can say we'll study about the proper cell. Now secondly, what happens is, Golgi derived vesicles, okay, with microtubules carried along, and they accumulate at the center and fuse to form a sulfate. For example, see, for example, you can say this is a plant cell right now. Okay, these are known as Golgi derived vesicles. What they are doing, suppose these are the organelles of plant cell, these two. Okay, uh, these two are the organelles. Okay, so now what happened is, they started to get condensed, and later on form a cell plate in the middle, which leads to the partition of the two cell organelles. And furthermore, what happens after this cell plate get matured enough? Okay, it starts to break from the cell from middle to outward, middle to outward, towards both side, or like let's turn into 180-degree. Okay, suppose cell plate is formed here. Now this one is plasma membrane, this one is cell wall. Okay, cell plate gets touches to plasma membrane and fuses it with. After the fusion, what happened is, they try to break the cell wall into two. We can term this as plasmodesmata. Okay. Then at last, after the connection with the cell plate, cell wall also, the cell divide into two. It is the last connection between this cell, and after that it breaks into two, which each of has this new plasma membrane and cell wall with organelles inside it. Basically, in both the system, division only happens at the mid zone, we can say equator of the mitotic spindle. In animals, it forms at the contractile ring. Plants, we can say cell plate occur, okay, at the spindle midzone. Then division happens. But here we have to consider like it done outward to inward, and vice versa here, which is inward to outward, or we can say middle to outward towards both sides, like this. In this case, this is this. Now we'll talk about binary fission in bacteria. So basically, what is binary fission? This literally means division in half. Division in half. It is a primary method of reproduction in prokaryotes. And say primary method of reproduction in prokaryotes. What are prokaryotes? We can include bacteria and archaea. Bacteria, archaea. Okay. This is done asexually, which means no involvement of gametes. A single parent divide into two genetically identical daughter cells. Genetically, but are identical, nearly same daughter cells. Sometimes, like single celled eukaryotes, for example, amoeba, we can say, binary fission occurs also, but mitosis is also involved. But in the case of bacteria, no mitosis involved in bacteria division. Basically, how it is done. For example, you can say this is a bacteria cell. Okay. And this layer outward, we can call it cell wall. Inward layer, we can call it plasma membrane or PM. Then there is one circular DNA inside. Okay. We can call it bacterial chromosome. Okay, also the DNA is also attached to the plasma membrane here. DNA also attached to plasma membrane, so it can be fit inside, and also it is super coiled. You can see it is coiled, so it can fit inside the cell, and folded, because like in the example in the E. coli chromosome, we can see E. coli bacteria, when the DNA is stretched out. Suppose this is E. coli bacteria, here's the DNA. When it's stretched out, it is 500 times larger, or you can say longer than the cell. The DNA is compacted with DNA binding proteins. You can say DNA binding proteins, just like histones, we can say in the eukaryotes, just like histone. I'll talk about later about histones. Eukaryotes. Now what happened is, there is a thing called origin of replication. It is a specific site on a chromosome when the DNA copying starts. For example, this is a DNA. Okay, we can term this site from this end to this end origin of replication, or OR. The DNA copying start here. Now how it is done? The replication is done toward both side, opposite end, from one end to the left, we can say, and to the right, from both side. Okay, for in a cell, you can say, for if the DNA chromosome if the replication start from this end to this end, it will start from here also and to there also. Okay, so it means it has two origin of replication. As we see, moving on to further, for example, like at this end the replication OR starts. Okay, now this origin, we can say, is first end, this can call second end. This will move to this side and replicate this area toward the end, we can say opposite pole. Okay, and this will do same. Okay, toward this end. Furthermore, while the cell division is happening here, the cell elongates also. Okay, it stretches so that DNA can be replicated further. This is done due to a protein called actin-like protein. We can call it as PAR-M, as per the scientists are researching, which help into movement of DNA origins, we can say origin of replication. Okay, and second is tubulin-like protein, FTSS, as per the scientists are researching right now. What does this thing do? This form a ring in the center. formation of ring like structure, we can say ring in center to division in cell to indicate, or we can say start division in cell. It is observed by fluorescent microscope. So basically, like for example, this is a bacterial cell wall. It's plasma membrane inside. It's DNA. We can say this point to this point, original of OR, original replication. Then what happens? This get replicated. Now there are two origins. One is here, one is here. Okay, two copies of origin of application here. Now it's basically continues again and resulting in cell elongation, cell elongated. Okay, one origin here, second origin here. Okay, then what happen next is it forms a groove like structure, like just similar to in the animal cells. And also, it like the plasma membrane, we can say it gets pinched inward by tubulin protein, which was FTSS FTSZ protein. It pinches the cell wall into two. Then at last, two daughter cells form, okay, with a separate DNA chromosome material inside. Now, in comparison to the prokaryotes and eukaryotes, there are some things like DNA structure for prokaryotes, single circular molecule. Okay, multiple linear molecules. In eukaryotes, you can say no nucleus here, nucleus present, no spindle apparatus, the hair is present. Mechanism was DNA attaches to plasma membrane. Okay, cell elongates, divides by membrane constriction. You can say pinching of cell wall. We can say pinching of the plasma membrane inward by a protein called as tubulin, which was FTSZ, and also in here chromosome attached to spindle fiber, which are separated by mitotic spindle. Separation occurs. Similarity, result of two identical cells. Which key proteins are used in the prokaryotes? Tubulin like FTSZ and PARM like actin. Actin filaments. Okay, and in the eukaryotes, tubulin, which forms spindle fibers, and actin, which forms cytokinesis ring. So like why does binary fission is necessary? First, it is rapid, done very quickly. You can see E. coli can divide every 20 minutes, okay, which resulting into exponential population growth. And what happened is it allows bacteria to adapt quickly to mutations. As you know, like the mutations in one generation can spread rapidly, can spread rapidly. Moreover, understanding of this help in formation of antibiotics. Understanding of binary fission helps in antibiotic development, as because what does antibiotic do? Like they break the cell wall of the anti for the like bacterial cell, and then target protein like FTZ, and then the problem gets finished.